My name is Ian Smith, and I'm a graduate student in the Hyde Cadence Lab at the University of Toronto. Today I will be presenting work on similarity metric learning in perturbational datasets. So perturbational datasets measure changes in biological features due to perturbations, like compounds, genomic perturbations, or disease states. An example of a perturbational dataset is the L1000 project from the Broad Institute that measures changes in gene expression as a result of treatment by various reagents like drugs or genomic perturbations in cancer cell lines. And this is the data set that I'll be discussing in this poster. Perturbational signatures can be thought of as a matrix where the rows are the features that you're measuring, in the case of L1000 gene expression, and the columns are samples, or again, in this case, cell lines plus perturbations. Perturbational datasets use replicates as a way to both gauge the signal to noise ratio of the measured process and assess the technical quality of the assay. This is data from L1000, where you can see that on the x-axis there's cosine similarity, where farther to the right it means the signatures are more similar, and replicates are shown in red versus all pairs of signatures in blue. And as you can see, the replicates are more similar to each other than random pairs of signatures. But that's not the only application of replicates. The technique that I use in this project, similarity metric learning, is an application of self-supervised learning, which is a technique from machine learning. Self-supervised learning has been successful in fields like image representation learning and natural language processing. And in this technique, an embedding is learned in the space that brings samples that are a priori known to be similar closer together. So in this case, two handwritten signatures that are known to be the same or known to be written by the same person are learned, the embedding is learned in such a way that brings these points together, and signatures from two different people are learned to be far apart in the embedding. And this technique has been widely successful in recent years. So the technique that I'm applying here leads to a metric that I call rectified cosine. And this is a parametrized similarity function. Your ordinary cosine similarity between two signatures A and B looks like this. It's just an inner product scaling by the norms of those metrics. Rectified cosine introduces this linear transformation on the data or on the signatures, and there are parameters in this linear transformation. We can then fit those parameters using something like mini batch gradient descent to use the replicates to learn an intrinsic metric on the space. And so the net result of learning this metric will be to push this replicate similarity to the right. Conceptually, rectified cosine identifies intrinsic axes of variation from the data and then scales them in such a way that replicates are maximized. The similarity between replicates is maximized. For instance, it could be that certain biological processes or pathways are either over or underrepresented in the feature space of choice for the perturbational data set, leading to an inconsistency in how the data is represented. By adjusting this, rectified cosine fixes and improves the recall on the original data space. So to benchmark this, the technique is to take the signature of a compound, compute the similarity to everything in the data set, and then rank all the signatures based on similarity and look in this result list to see where other signatures of compound A are, with the assumption that two signatures of the same compound should be similar. That leads to this result, where the rank of the replicate is on the x-axis, where 0 indicates that it's the most similar and 1 indicates it's the least similar. And the y-axis is a cumulative fraction. What you can see here is that the rectified cosine variations, of which I'm showing 2 here, trained on 2 different cell lines, shown in purple and orange, outperform the other kind of standard similarity functions that you might use off the shelf, including a GSEA-based metric, cosine, Pearson, and Spearman correlation. This is in the ASC cell line. What this shows is that this learned metric can improve how replicates are how similar replicates are to each other. But that's not necessarily that surprising because that's what the data was trained on. So to provide another benchmark, I look at pairs of compounds that belong to the same class. So these compounds in the same class that have the same mechanism of action, they might target the same genes or pathways. And what you see is that these weren't used in the training of the metric at all, but rectified cosine still shows an improved recall of same class compounds compared to cosine. And at a particular false discovery rate, for example, 5%, rectified cosine shows an improvement of something like 30% of the recall of standard cosine. So this shows that not only is rectified cosine learning an intrinsic metric on the space, but it's generalizing to data that it wasn't shown in the training process. So to summarize, similarity metric learning is a powerful technique to use that uses self-supervised learning to learn a dataset specific similarity function. It improves the recall of known biological associations. Importantly, it embeds the data in the original feature space, so you can still use techniques like gene set methods. And it can be applied to a host of other problems, like clustering, that use similarity-based functions. Thank you for your attention.